Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in previous life I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aiming the public with the tools of biotech.
My name is Faz, I have a PhD in microbiology, I'm mostly working in bacteria, but I've also worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen this week.
And towards the end of the month, we'll be doing a deep dive into a really interesting microbiology paper. But if you have any suggestions for papers that you want us to do a deep dive into, please tweet us at microtwjc if something's caught your fancy.
Yep, you can follow along with any of the papers that we discuss in our Shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you, so please use the comments or tweet us at microdwjc. Yeah, and boy, do we have a show for you today. We're looking at some of the first data to come out through on the clinical severity of Omicron. We also have some data on how well it does against T-cells, and also a look at how SARS-CoV-2 can spread from cell to cell. But wait, there's more. We also take a look at other stories from microbiology, such as how some antibiotics might actually help bacteria become immune to phages. We also have a protein that makes bacterial membranes go all noodly. And finally, we take a look at how one species of algae that might have survived the extinction of the dinosaurs.
Yep. Pretty excited to talk about this stuff today, and we're going to kick it off as usual with our coverage of the SARS-CoV-2 pandemic. Particularly this week, we're seeing a bunch more papers telling us a little about this Omicron variant that everyone is quite concerned about.
Yeah, and so we're starting off with this paper from South Africa. So this is coming from the group that discovered and were the first to really publish about it. And it's taken a couple of weeks for the data to come through, all the while other people have taken bits and bobs. So this is looking directly at about the epidemic in South Africa specifically, and in the Gauteng province, as I believe it's supposed to be pronounced.
Yep. Yeah, so I think we'd seen very similar stuff to this before, but this is a different group, and they're really using that S gene target failure as their marker for Omicron.
Yeah, so for a quick refresher, S gene target failure is when you're trying to sequence for the spike protein, but it drops out, and it drops out because of a specific set of mutations, which is the delta 69 to 70 in one part of the spike protein, which would be bad, except people usually in most tests they test for multiple genes across the SARS-CoV-2. So when you see this knockout, you know that mutation is there, which is an indicator that this is Omicron.
Right.
So it's quite a nice way to actually track how this is spreading.
Yeah, actually, it's quite like it's almost lucky, right? Because I think the S gene target failure was originally identified during like the alpha wave or whatever, and then
that's right.
So like that variant too could be the thing that's being detected, but of course like what's circulating now in most places is the delta strain, and so seeing that increase in S target gene failure, and attributing that to the Omicron, like it's sort of like there's a nice background, right? Of like there were no S gene target failures for some time, and now we're seeing them pop up, so they can use that as a great way to track the spread of this variant.
Yeah, and so this paper they contextualize it in terms of the other like big outbreaks that South Africa has seen. So you've got your first like outbreak, which is the original recipe SARS, and then you got your second one, which was the beta variant, which was very prevalent in South Africa, and then finally you get the Delta variant, and just in the past month we've got Omicron.
Yeah, really nice visualizations. Like I feel like that's so easy to read, that graph with the different colored bumps in daily cases.
Yeah, and I also like kind of want to draw attention to the fact how steep each curve is. So you can kind of see that Delta is much steeper than Beta and the one before that. That could be due to testing, but then you see like Omicron, it's just really spiking up really high, and that kind of tracks with what we've been seeing in other countries. So I know I think it's just about beaten out Delta in the UK. It's like, and we're seeing record case numbers here. And yeah, you can see like a more detailed timeline of like how it appeared, and you've got like a little map of Gauteng province, I think, and So you get so yeah, I think the visualizations of this are probably quite easy to grok, quite easy to understand.
Totally. um, Yeah, and they go into looking at some of the evolution, which nothing new here, right? Like this is sort of what we saw in some of the earlier evolution graphs in Figure 2, right? Like
Yes.
Omicron pops up late 2002, and it has this, right? There's many different things that have been sequenced at this point, but if you look at like, if you track sort of its chain back, there's not a lot of isolates that come, like, that are fleshed out in like the earlier periods of time. So this is, I mean, we've said this before. Maybe many people have heard this on the news now, right? That probably it's evolved somewhere where there wasn't a lot of surveillance,
Mm.
and then it appeared in localities that did have surveillance. So that's why we don't have some of those earlier isolates from its particular branch.
Yeah. And they do like talk about the Omicron lineages because Omicron itself there is some diversity in there. There's
Hmm.
BA1, BA2, and BA3. And I think they're seeing that BA1 is the one of these variants that's winning out of there within Omicron.
Hmm.
It's spreading out, so that's spreading internationally.
Yeah.
Um, and they do some things like trying to check whether maybe like things like looking at whether there's a recombination event that could explain why this looks so different, but they didn't find any convincing evidence of that.
Hmm.
They also like looked at other ideas of like maybe it was like sequential mutations. I think that's more likely.
Hmm.
Um, but yeah, overall, I think that this is it's good because I think it's important to credit this group as well
Yeah.
for the finding. Um, and it does like put the data in quite a nice
Yeah,
way that's quite easy
I
to
like the
understand.
packaging. It's nice to see like a good overview because also, when we first covered this maybe like 2 weeks ago, like we were just picking up little scraps of things from all the papers that were coming up, but there's a nice story here, I think, about Omicron. And yeah, so this is that other piece of the puzzle, right? The one that most people are concerned about, the spike protein, and seeing the mutations that are um, all across the surface there. And yeah, these mutations, right? We don't have solid evidence on all of them, but there's sort of the implication that many of them will be contributing to immune escape. So the antibodies being less effective in binding.
Right. I think that brings us neatly to our next paper, which also comes from South Africa, which is early assessment of the clinical severity of SARS-CoV-2 Omicron variant in South Africa. So this is one of the first papers to actually look at and ask the question: is this actually more severe than previous strains?
Oh yeah, wait,
Um,
1 second. Let's try to address this question in the chat. Is this similar to 1918 flu at year 3 being less severe? Right. So I think this is sort of speaking to, and I think we'll come to this for
mm,
sure in some of the later papers, right? Like that we have more clinical evidence now to see like the severity of it. But I think the papers that we're going to cover, it's in light of vaccines. Um,
mm.
and that's not so like, and I also don't really know anything about this history that you're talking about, Jackie. Like, if you could maybe, if it's, I don't know if you have that information on hand. Like, I'm not sure about what happened in year 3 of the 1918 flu. Like that, it would be, that's sort of there's certainly the idea that if a virus is becoming endemic, right, like it can't kill its hosts. So there should be
Well,
some trend towards that. But I can't say anything for sure. Sorry, Fuzz, you want to say something?
so yeah, what I want to say is that so I'm not familiar with the historic situation. All I'm kind of familiar with the generalities that like the 1918 strain eventually dominated, and lots of people had pre-existing immunity, and then that, and I think like the vaccines are almost performing that function of
Hmm.
pre-existing immunity that was happening there, where like so in I can't really speak to the UK situation, but what we've seen is in this latest epidemic wave, I think for Delta that the number of cases and number of deaths have kind of become decoupled from each other a lot,
Hmm. Hmm.
where we're seeing quite high case numbers, but the number of like the relative amount of people ending up in hospital as a result is decreasing,
Right.
and does and that is probably because of the vaccine. Because again, we got very hardcore on the vaccines here.
Hmm.
Um, so that is probably quite a strong reason for some of that pre-existing immunity.
Yep.
Um, And Omicron, I think we've seen in previous papers that it's slightly evading that vaccine immunity, but the question is, how does that actually contribute to severity, which we're going to be looking at the next two papers we're going to be discussing right now. So
Right. Yeah.
keep watching.
Yeah. Okay, that's great. I think that's a good way to address that question. Yeah, and so this is exactly that, right? Or is it very similar? They're focusing now on the clinical severity of being infected by Omicron, not just whether people are getting it, but are those people ending up in the hospital?
Yeah, so this pulls in data from a number of different places, from the National Institute for Communicable Diseases. They've got like notifiable medical. They've got a bunch of surveillance systems in South Africa for dealing with this, and they get case and test data. And what they're effectively just looking is just the number of people who have confirmed Omicron or had that the S
SG
gene
target failure.
dropout,
Yep.
and seeing whether how often they end up in hospital.
Hmm.
And it's a bit encouraging. I think I don't want to break from my usual doomer mindset here. So
Yeah,
because
so
I've got a brand to keep up.
yeah, so they show again, just as in the previous paper, that like they're seeing this surge in S gene target failure dropouts. That's correlated with what we think is the Omicron variant, right? Kind of spreading through everywhere. And then they then they just look at people in the hospital who have this S gene target failure, and they try
Yeah,
to say, yeah, what's the odds ratio that they end up with severe disease versus those that don't have S gene target failure.
yeah. So and they do some comparisons with the previous outbreak they had with Delta. I think that they use that as their control
Mm,
and to compare like how bad it looks. And I think that what they tend to find is that things look a bit better than in the previous Delta wave.
mhm. So of course there you also have to think that vaccine rates have gone up, right, between
Yeah,
those 2 points. So it's like not this is not necessarily intrinsic about the virus, although it could be as well. There's also that covariate, right, um, of vaccinations.
yeah. I think that the key thing is that this is like the first pancake analysis where like it's not going to be perfect. This is
Hmm.
they there are lots of covariates and not necessarily able to give us an idea about like such as the amount of vaccinations, like how much of these people who aren't ending up in hospital have vaccines versus people who do,
Hmm.
and also people who've had had infections before that.
Yep.
We don't really get much data on that.
Right. But
Um,
they do. So like I'm saying, like in that bulk idea that maybe there's less people ending up in the hospital, that doesn't take into account. But then they do have like a line in this graph saying SARS-CoV-2 vaccination, and so that's just for those who end up in the hospital. Though, how well do people fare if they have the vaccine versus if they don't? They're just saying people with the vaccine,
yes,
they do fare better. They use the no as the reference point. So
yeah. So I think that I mean it looks good. I think that there's still a lot of work to be done because again it's only a month in, and sometimes it takes quite a while for like deaths to show up from these initial infections.
hmm.
So we're doing our best to predict what will happen based on kind of fairly scant data at the moment.
Hmm. Oh yeah, comorbidities. Yeah, if you, I mean, it's knowledge that we already have, right? Like if you have hypertension, diabetes, chronic cardiac disease, chronic kidney disease, asthma, chronic COPD, malignancy, HIV, or other or active or past tuberculosis, you're gonna fare worse again, right? So like, this is stuff that we know, right? Like, I mean, I don't mean to chuckle
Yeah,
like that, but like, yeah, this is stuff that we know. You don't want to be infected if you have those comorbidities. We all sort of have to work together to make sure that we're not just spreading it through the population, right? Then it hits those people with the comorbidities quicker. Um, yeah, but again,
yeah, actually,
but
yeah,
sort of positive in that uh, although there's this huge explosion of infection, right, which we need to control because we don't want those people, like, right, folks that would end up in serious disease conditions to end up in the hospital. There is some, I guess, like the silver lining, right, that uh, the
yeah,
vaccines are working, or like, we're if you do end up getting Omicron, right? Like, hopefully your chances would be better, statistically speaking.
Yeah, I think that that's something that we. It's hard to say that whether the virus itself is intrinsically more or less infectious or like less severe at this
Mm-hmm.
point because we've got this massive confounder over all this pre-existing immunity.
Yeah.
And the next paper we're looking at, well, it's not a paper, it's more like a report that tries to answer this question
Mm.
through using quite a lot of maths actually, which is unfortunate, but we'll try to get through it.
Yeah.
So this is not peer reviewed. It's a report from Imperial College London through from some of the people who are running the advice to the government on what should be done. And so they again, this is a bit more simple because of course it's got a more unified system. So they've got like NHS and immunization. They talk to each other, so you know when someone has got when they turn up to hospital whether they've been vaccinated or not,
Yeah.
and maybe also whether they've got a pre-existing infection.
Yeah. They also know
Um,
what vaccine they got in this sense. So like they're
yeah,
stratify out their analysis by what vaccine, the time in which that they got those vaccines.
yes. So we got a lot more information, but because it's very early on, this paper they're very understanding of the fact that they don't have perfect information. So instead of trying to go with what they have, they fit it to a model to try and predict what the real situation is based on the data they already have.
Mm-hmm.
Um, so yeah,
But
so
their
that's why we got
takeaway is sort of the same as the previous one, that the incidence of severe infection is lower with this as compared to Delta, right? Like lower as compared to
yeah.
Delta in those people who are vaccinated.
Yeah, and I think at this point, because of the stage they caught this infection, they were able to catch Delta infections at the same time as Omicron infections because at this point it was in a period of transition for the UK. Um, so instead of so I think that because I think they do try to like say that they do a bit more with this paper. I think they look at like reinfections as well because I think another thing is that Omicron does. I think they see here is that people who've been vaccinated are more likely to catch Omicron than vaccinated people would be to catch the Delta variant. Let's say.
Mm.
So I'm not saying that.
But like again, there's the covert of behavior there, right? Like maybe people are. It's like the behavior of people to catch Omicron versus Delta. I'm not sure. Like that's just not addressed, right, in this type of analysis.
Yeah, it's very hard to address the behavioral aspects of these. I think
Mm-hmm.
we've seen that done before where they overlay like Google motion maps on things of like where people are. But
Mm-hmm.
even then, you're only looking at a very general picture. You can't get an individual level because I don't think anybody wants to give that kind of data away.
Right.
Um,
Right.
yeah. I think so. This tries to like take into account the the data of like past immunization. I think point out that that is just a big confounder at the moment. That means that they can't get a perfect idea of what Omicron would be like in, say, an unvaccinated person. But
Mm-hmm.
what they seem to be saying is that clinical severity tends to be lower in people who so compared to the Delta. Um,
Mm-hmm.
so I think this. So I mean, I'd be interested to see what the effect is in unvaccinated people compared to Delta. I think that's the space where we're going to find out whether Omicron is intrinsically more severe or not.
Yeah,
But
but we
yeah,
already have really good information to suggest that like people who are unvaccinated aren't going to fare well. Oh, I see. But you're saying to understand something about Omicron
yeah,
itself, we would want you'd want to see the comparison. I see.
yeah. I
Yes,
mean, I
yes.
would want to see the comparison from an ethical perspective. I don't really because unvaccinated people should
Right,
be a diminishing resource at this point. Like,
right,
um,
right. I mean, that's if the public health initiatives are doing their job, right? We shouldn't have that. Like, it would be basically difficult to have that information at some point. But from the scientific, like, if you wanted to know something intrinsic about whether Omicron caused more serious disease, that's the proper comparator, right?
yeah,
That's what you're
that's
saying.
what I'm saying. And I'll be honest, I
Yeah.
don't need to know it that badly. So I'm happy if we never get enough numbers of people dying to. So
Totally.
yeah,
Yeah. I totally agree with you. It's sort of like we want to see the exit of these things, and all this information is supposed to be helping um, I guess decision, right? Like policymakers, like think about like, oh, like what does it mean in terms of like should we be dialing up our like non-pharmaceutical intervention stuff? Like should we be dialing up like restrictions on whatever people
Hmm.
think is like the right intervention? And so it's sort of, I guess, in some ways, like this data is trying to tell us like we we can continue doing what we're doing and we shouldn't expect to see some sort of disaster. It doesn't explicitly model that type of case because if you remember, I'm just remembering like earlier things that we've seen in the pandemic, like sometimes they'll explicitly model cases
Hmm.
of like what if we keep doing what we're doing versus what if we try to change something up to impact that. And like we're not, that recommendation is not explicitly coming through here, but perhaps that is what we could sort of, that I think that that's where the authors are trying to get at ultimately here, right? That if we continue to vaccinate folks, we don't have to worry about increasing hospital burden.
Yeah, but I mean, at the moment, from what they see, I mean, even the data in people who are unvaccinated, it does seem to be less severe than Delta. But then again, this is still early
Mm,
days,
mm,
and I haven't got
mhm.
my full critical eye on this. So um,
Right, right, right. This is just what we're getting from the skim.
yeah, this is just from the
Yeah,
skin. This isn't like a full review of this, unfortunately. I wish it was, but yeah, I
yeah,
think it's still good news. Is something that's rare these days, and it'd be nice to be able to stick by it and say yes. But I mean,
right,
I
right.
do want to like, but then Omicron is very much more transmissible. So even if it is much more severe, more people having it means you've got a bigger pie. And might be a small slice of the
Yes,
pie, but if the pie's bigger, you're gonna have a lot of cases to deal with.
yes, yes. Yeah, that's a very good point.
Um,
Okay.
yeah, so that's something for public health people to kind of think about of how we distribute hospital resources. Um,
Mhm.
and your choice of whether you want to take that, put that kind of stress on people. I don't know. I don't know. I can't advise what
Yeah,
people do.
yeah.
And we're not doctors. We're the other type. So apologies. No, ask your medical advice from doctors or people with proper qualifications. I just mess around with bacteria in a petri dish for like a couple of years. So I mean,
Totally. Yeah, let's move on to the next paper. You picked
yes,
up Omicron-specific cytotoxic T cell responses are boosted following a third dose of mRNA COVID-19 vaccine in anti-CD20 treated multiple sclerosis patients.
yeah, this is an interesting one because I think the thing. So why this core cohort? Why anti-CD20 multiple sclerosis patients?
Yeah.
Because they tend to have very suboptimal antibody responses. So understanding whether I think CD20 knocks down the antibody response in them to try and
Yep.
ameliorate the
Yeah, so
symptoms.
this is sort of trying to do, it's trying to do a with a natural this population of people exists, right?
Yes.
Multiple sclerosis, chronic disease, sort of very complicated and potentially confusing. Part of it is unknown mechanism of mechanism of pathogenesis. It's related something to the immune system and that immune system hurting the nervous system of these individuals. And so there's like a whole bunch of different therapeutics that exist in this space, and one of them is anti-CD20 antibodies that essentially suppress the immune system response in such a way that empirically it's been determined that this is quite good for the outcomes of multiple sclerosis patients. But what that gives scientists, I guess, is a window into a population of people that have a very specific, right, like an anti-CD20 antibody. That's almost like a research like tool in some ways. It's being used as a therapeutic tool here, but it's also like a very specific way of hobbling the immune system. I think it's like in B cell T cell interaction. It prevents some sort of like interaction there that like trains the B cells in a certain way. And so the question gets asked, everyone's getting vaccinated because that risk is much higher, right? Like in terms of like there's the risk of COVID-19 infection. So everyone's getting vaccinated. And so let's look then in this specific population that have, for therapeutic reasons, a defect in their immune system, and then how does that affect their response to the vaccine?
Yeah, and I think this population, you really want to know whether the T cells can protect, because that is most of their response to it, right? That's their main line of
Right,
defense.
right,
And I think this.
right. And, oh yeah.
Oh yeah.
Sorry, I just want to say like that's great. And right, this is in light of when we say like Omicron is immune evading, right? Like it's not responding to the antibodies that we have. In some ways, that's what's happening in these people, right? Like I would say like Omicron doesn't even matter in this sense. Well, maybe it does just because people want to know like there's other changes on Omicron too, right? That maybe people are afraid of. But you could just see this as a way of looking at what happens when SARS-CoV-2 doesn't have like any pre-existing neutralizing antibody complement in a body, right? And the vaccine is still working through some other mechanism to suppress serious disease, perhaps.
Yeah, and I think there is some element here of like, because I think this was important for the Delta outbreak and the previous variant outbreaks, and this study was started before Omicron was even in a twinkle in someone's eye at this point, where it was started in March 2021. And they recruited a bunch of people, and they were taking like blood samples, where they get T cells from, and through that they can actually see like they could get. So I think they got people who are vaccinated with who are vaccinated or and then they tested to see whether they had T cells that were specific against the vaccine, the Delta strain, and the Omicron protein to see what. So they had this thing called AIM, which is activation-induced marker assay, where you essentially show T cells an antigen and see whether it makes them angry or not. And if it makes them angry, it's probably that's my scientific way of describing it.
Yeah, I think we covered AIM in another episode. Now I'm when you said it now, like, oh my god, like we talked about that because right, like there's like a suite of like cells when they get immune cells when they're exposed to something that makes them angry, they turn on a whole bunch of different genes. And traditionally, the way that you would study that is you would just like take those specific genes and say like, if this gene is turned on, then that means that there's some sort of activation event. I think AAM somehow like condenses a bunch of those into one. I'm not quite sure, but like
I think so. We definitely covered a T cell because we covered a couple of T cell papers that I think use this, and that's why I think
something about epitope. I think when we were talking about something like a T cell epitope specificity, the readout was also AIM plus.
And
man, you can tell like this is like the immunology where we've like learned immunology just to talk about it, and then like several weeks later, months later, it's kind of gone from my memory.
But yeah, I think the epitope mapping is an important point because what they did here was effectively the same thing as that, similar to that, where they took the spike protein, chopped it up into 15 like length proteins that are enough for the T cells to like grapple onto, and then check what the T cells respond to. And I think the key thing here is seeing whether they could respond to as well to pools with the Omicron mutations or the Delta mutations.
Right. And it's kind of the same, right, is what the takeaway of these are.
Yeah, they're kind of. There might be a sensation that there might be some slight like lowering, perhaps potentially, but yeah, I think the key point of this paper is that post boost, their reactions are much better to all of them, and
another reason to be boosted. So like. Oh, actually, that's interesting. So boosting not only would impact, right? Because like I think the narrative we were saying last week was boosting is gonna boost those neutralizing antibodies, right? So then maybe that infection will never get a toehold. But here, boosting is also having an effect on the T cell reactivity to Omicron.
Yeah, so it produces a lot more CD8 T cells. So that's those T cells that naturally I was about to say natural killer, but that's a different cell. They're the cells that kill off cells that are infected with the virus. So they hit the virus. When those cells get turned into virus-producing factories, these T cells are there to destroy them before that happens. And yeah, I think they don't necessarily see the same thing with CD4 T cells. I think the CD4 T cells more or less stay the same after boost, which is interesting actually, because I'm not really up on the immunology side of how boosting works, but yeah, I think it does make sense for the effector side to be boosted up rather than the kind of the brain side of it. I see the CD4 T cells as almost like the like kind of organizing factor for the response.
Right.
But yeah,
Yeah, interesting. That's quite short paper actually, right? Like just two sets of observations using this particular view of into the immune system
yeah, yeah. I mean,
in positive,
it's
S specific. Yeah,
again, this is like quite good news. I mean, we're hoping for the fact that the T cells response wouldn't like be too evaded by Omicron because of the fact that the way the T cells respond to it means that they can react to many more epitopes. I feel, and they can do a lot.
yeah,
There's a lot more freedom with the T cells to react to stuff that Omicron can't change without screwing up its
right.
function.
Yeah, yeah. Good news in light of the immune
Hmm.
evasion aspect of Omicron, right? And so like that's for folks who like yeah for immune evasion that immune evasion aspect, right? People we saw from last week, I think, right? Like the antibodies are a lot less effective. So there are people that don't make antibodies. They have a different type of boost response to the vaccine, and presumably that's also happening within people who can also make antibodies. So I guess an important reason to get your booster. But once again, like not helping people who don't have access to those
Yeah,
things.
that's right. I mean,
Yeah.
unfortunately, yeah, it's
Yeah.
quite because I live in a country which is where the government is literally going to bang down your door and like politely ask you
Right.
whether they and like they'll have a vaccine for you at your doorstep, like Deliveroo.
Right.
Um, so
Yeah.
yeah, so
But interesting. Yeah. This is important information to have like that. Um, hopefully also maybe makes people feel comfortable like or encourages people to get the vaccine, right? Because that's I think something that we want to move the needle on.
yeah, I think so. Oh, move needle. I see what you did there. Um,
Okay.
come
Shall we
for the
move on?
science, stay for the puns. So, shall we move swiftly on to the next paper, which is
Yeah.
SARS-CoV-2 Omicron variant replication in human respiratory tract ex vivo, a paper I've been so excited about. We talked about the press release of this paper last week,
Yeah.
and now they actually have
We just saw three images on a website. We were excited to talk about it, and now we get the preprint.
yeah. So in this case, what they did, I think they grew up some SARS-CoV-2 in TMPRSS2. I think they used they were for testing
This is
sorry.
no, no, it's good. I just want to say this is ex vivo, right? This is like they have tissue samples from these specific human locations, so lung tissue samples, bronchus tissue samples,
Yes.
and so the model. I think the reason why we were excited last time and continue to be excited now is this model is great because we're taking the tissue of the most relevant tissue and studying it, right? Of course there are caveats because it's not within the full context of the organism, but we're still a lot closer, right, to the full organism. And so the data coming back from this, like, presumably is going to give us like that much better insight into specific pathogenesis.
Yeah, yeah. They test like the bronchus. I think they specifically say it was from non-cancer cell, non-tumor bronchus, obtained from patients 51
Mm-hmm.
to 78 years of age, and they grew them up in like ex vivo. So that means they took out these slices of meat. I don't want to say meat. Like organ, and grow them up in the tube,
Yeah,
and they
yeah, tissue.
tissue's the right word. Ah, gosh. All my lecturers would hate me for saying that stuff like that, but um, yeah. And they added just adds a little bit of Omicron, so I think they got it from a Hong Kong patient. And they checked the replication of Omicron at like 24, 48, and 72 hours. And they like
Mm-hmm.
looked at and they saw like that it grew much better in the bronchi than compared into the lungs, which is quite interesting. Um, uh,
Yeah, this was the result that we saw last time. Here we're just, I guess, it's like here's the data, right? Like
yeah,
really
here
full
we
force.
yeah. Let me see what because I think they also had some like cool stuff on furin cleavage as well because I think they found that in oh, I hate the zoom function on all scientific journals because it never does what I want it to do.
Yeah, it's like resizing in a funny way, but
Yeah. Um, so I think they found like I think ACE2 cleavage was different in these different locations potentially.
Mm.
Um, and so like I mean, I think they also find that like they use camostat. So camostat is this thing that inhibits cleavage. So the
Mm-hmm.
and see what like, the effect that that has on I think or on the amount of SARS-CoV-2 in these areas, and they found that I think
Yeah.
like I like
I think it's also about tissue distribution of ACE2 in
yes
this figure, I believe. So like the bronchus also just has more ACE2, and so what this dovetails with actually is something I think that we were talking about. Like these mutations might make them is the mutations on the spike for Omicron make it more likely to escape antibodies, but also might have some effect on its ability to bind ACE2. But in a tissue that has lots of ACE2, that's not a
hmm.
problem. And so that idea that it's like, yeah, that it might be worse at binding but better at fusion. I think that was the thing we were talking about some time ago. Like that's maybe that I think that that's what the authors are trying to drive towards in terms of hypothesis of like this is why we're going to see that's why we're going to see Omicron like preferentially infect this tissue type, the
Yeah,
bronchus.
it's interesting
um,
because I think it's complicated by the fact that by what they find is that it's. I mean, I think a couple of papers found that it's not
there's
quite as good as being cleaved than say Delta, but
mm-hmm,
Delta
mm-hmm,
is super cleavable. I think that's the one thing we found. Like the Delta's like main app like kind of advantage was the fact they were so, and I think that's kind of demonstrated here with the fact that they add this chemistry stat, which blocks like cleavage, which kind of
mm-hmm,
completely shuts down Delta. Uh, whereas like
mm-hmm.
with Omicron and other like strains, it's like the other strains have other kind of things that they can resort to spread through in cell culture.
Right.
Whereas Delta's kind of
Mm-hmm,
everything's
mm-hmm.
put into like being good at being furin cleaved.
Mm-hmm, mm-hmm. Yeah, like a really different. I maybe that's like just also really cool. I think it is from the evolution standpoint, right? Like the strategy a virus can use to be successful, because we're seeing that relate. Like we see the effect of that in the fact that there's these waves coming through our population, but on the molecular level, like there's actually really it's quite different, right? Like Delta is leveraging this ability, a certain ability to drive the wave, let's say, but Omicron isn't. It has some other ability, right? That's it, right. Yeah, produce the wave in society.
Yeah, I think when skimming this paper, I wasn't quite clear on exactly what the benefit Omicron was having in the bronchus compared to the lungs, or whether there was something in the lungs that intrinsically made it less able to infect. I
Mm-hmm, mm-hmm.
So I don't know whether you.
I did. Yeah, I don't think I. I don't think I picked that up either. But it is more effective in the bronchus versus the lung, right? That's figure one
Yeah,
in F. Like it is.
yeah. So I mean, I think we're comparing it to the other strains. It does seem to do better. So I mean, but I'm trying to like compare like how much better it does compared to the other strains. So looking at so it might so it and it's not so much. So it looks like not so much that it does better in the bronchus, but it just does much worse in the lung.
Does much worse than the lung.
Yeah,
Yes, that's what I would say
yeah. I
here.
think that's what this.
Yeah, because delta is also comparably doing well in the
Yeah.
bronchus. Yeah.
So so I get the impression that maybe like there's something in the maybe Omicron's more vulnerable to the immune system in the lung. Maybe there's something else in the lung stuff. But again, it's a
Yeah,
mystery. Could
doesn't
be
dive into
anything.
this in this paper, but like that's where your mind might go.
Right. Yeah. So
Yeah,
yeah,
cool.
really. Sorry.
No, cool,
Oh,
cool.
cool. Yeah.
Yeah.
That's all I have to say on that
Yeah.
paper. Next one is SARS-CoV-2 spreads through cell-to-cell transmission. So
Hmm.
now we're
Yeah. So this is an.
yeah.
Oh
Oh
yeah.
no, I was gonna say we come to the end of the Omicron-specific papers, and now we're looking at the SARS-CoV-2 cell biology papers.
Hmm. Hmm. Yeah, and this one too, it's not a news story actually. Like I think we covered I linked it in the Zotero if people know how to use Zotero. There's like a related papers option on every paper. And so I linked to the previous one that had the same finding essentially. So it's because the spike protein mediates fusion of the envelope to a cell, then it's not outside of the realm of understanding of biology that when those spike proteins get expressed on the surface of cells, that then those cells can just fuse together by themselves, irrespective of like a viral particle existing. And that's well documented in RSV. This it's a respiratory virus, and so the investigation would be, does this happen in SARS-CoV-2? And so they do this in culture, where they have they have a cell type that they transfect, so it expresses spike protein, and then a cell type that doesn't, and then they see, do they fuse.
Yeah. So I think like because I think they've seen some papers where they like look in vivo and they found like syncytia in like lung samples, and so that indicates that fusion does happen. I think here they're looking at things that are kind of more subtle than that, and they've got like these different setups in their petri dishes. So they've got cells that are basically transfected with this uh like I think pseudovirus that they use. I think they use two pseudoviruses. They use a lentivirus based one and vesicular stomatitis virus, and they got these. So one where they infect one group of cells with it, and they either mix them together with the other cells, or they separate them with a well. So where one cell group type is like in a well, and the other one is underneath, so they can't actually directly contact each other, and they kind of compare how well the infection spreads based on that. So the idea would be that when cells are in close contact, there'd slightly be better there'd be more infections because they've got that close contact, which gives this virus this other way of transmitting other than just being spewed out into the medium.
Right. So yeah, so it's still like I actually don't remember if in the other one they found it in actual patients.
I
Do you remember?
feel
Did you
like
pick that
I
up
picked that
from?
up from a paper, but I could be wrong. I mean,
Okay, okay.
it's not unusual for me to be wrong.
but but yeah, but like what you said about this setup is, yeah, that's, I like that observation. Like in some ways they're looking at the contribution of this particular effect to the, because they have because they're able to separate the cells, it means that they're looking at the contribution of the effect to the overall, right, infectivity in the cell culture. And that insight is more than just saying like it can happen versus it can't happen.
Yeah, and they're mostly they do use real SARS-CoV-2 at some point, but yeah, they've got like the cell free, which is just a cold culture, and cell to cell, and they
Yep.
do like compare the percentages of infection. So it's quite a neat look at that, and then they do a lot of complex like biochemistry where they try and like pick out. So I think they use a couple of drugs to try and like shut down different aspects of the cell to try and figure out how that has an effect on fusion.
That's right. Yes, and they then and through that process they tie this to like endosomal fusion, right?
Yes.
Where cells, right? Cells have endosomes, right? Like that's how they're like sampling stuff from this. That's how they're sampling stuff. That's how they're inside of the cells, right? They have vesicles and stuff. They all need to like merge at different times. Golgi apparatus, endoplasmic reticulum. You can tell this is just me
Yeah.
spewing out
So
cell
and
biology terms.
so inside the cell and eukaryotic cells mostly. I think that I think pretty much this is a eukaryotic cell specific thing where endosomes are
Yeah,
essentially like tiny little organs that transport important things throughout the cell. So it could be like nutrients, it could be signaling molecules. Sometimes it's just lipids to make sure each part of the cell is maintained, and it's a very complicated system that we're still trying to figure out the details of because it's there's we've only I think we've just like it's just become common enough that we have good enough microscopy skills to look at how each of these actions is going. Um, because I remember like so I came fresh from ACSE, which is like the I think
yeah.
American Society for Cell Biology is like which had like an entire track on endosomes. So I've just got that filled in my brain at the moment. Um,
Yeah, no, thanks for sharing that tidbit. So that these events are dependent. This cell fusion event, right, is dependent on those mechanisms inside of a cell. And then I guess the sort of the kicker question that they want to then, or the way they want to then relate that, I think, um, to immunity is that they try these experiments under in the condition where they add the serum,
right.
um, sort of immune serum to the different the different variants of SARS-CoV-2 into these cell culture dishes and see if that impacts any of this, the sort of the cell-cell fusion mediated infectivity,
Mm.
And it can't. So the thought the thought that they're trying to advance there is that this potentially represents, right, a way of a way, it's within your own body, right? This is not person to person, right? It's like in your own body, right? This is a way that SARS-CoV-2 can spread that is sort of not sensitive to antibodies. But again, it's probably still sensitive to T cells, right?
Yeah.
Cytotoxic T cells.
I think that like reiterates, I think, a theme that we've been going through. Antibodies aren't everything. They're an important proxy for immunity, but when you're looking at the immune response itself, there's multiple complex things. So I think there. I mean, yeah, I'm trying to, but yeah, I think that because antibodies might have something anti self cells. I don't know. Because I feel like that's why why it was important for the MS paper we're just talking about with so antibodies can respond to cells a little bit. I think
Yes, absolutely. Yeah, I
yeah.
mean, but those are, in some ways, those are also a type of autoimmune, right?
Mm.
Response, right? If the antibodies are binding to things. But yeah, so actually, I don't really, I mean, that it's an interesting finding, right? It's just saying that the antibodies that you have from immunity, like, they're not going to like coat your own cells and stop them from binding necessarily. I'm sure it also has to do with like the size of things. Might have to do with like what ends up initiating those
Yeah.
events. Yeah,
And
there are many factors that go
yeah,
into that.
and a lot of things antibodies do is target other cells, like to things. So like target phagocytes and other kind of like parts of the innate immune system things, and that probably isn't there in here in this sort of sample.
Right, right. SARS-CoV-2
But
spreads through. Sorry, I want to look at the related thing. It was the old paper was, or the paper that we previously covered about this was in the Journal of Virology, Hornich et al. Um, they infect cells. They're talking about cell fusion. They have TMPRSS. They use CALU lung cells. So that's a cell. That's a
lung,
cell
yeah,
line.
that's a lung cancer cell line.
Yeah. And yeah, HORN, but the O has an umlaut on it. Yeah, I don't think they don't actually find this, right? So I'm trying to, I want to try to tell people, like, is this happening in urine, right? When people are being infected by SARS-CoV-2, is it actually happening? Is there direct evidence for that? And I don't think that there is. I think that this is stuff that, right, we understand from RSV, which we do have direct evidence for, but we don't necessarily have direct evidence for in SARS-CoV-2.
Right. Okay.
But it's sort of understandable that it could be there. Yeah.
Yeah, I think that's yeah, you're right. This is a very indirect way of looking at it, but I think that yeah, the direct ways are quite complicated to find, I think.
Yeah. Or maybe we haven't looked for those specific papers as well, because again, what's the contribution? What's the contribution of a specific? What's the contribution of a specific mechanism of pathogenesis to the overall disease? Like that, those are really hard things to pick out. Maybe that's something that an animal model could be poised
Yeah.
to give us, right? If we saw it
And I
there.
think that
Yeah.
this paper is almost like trying to like hone down, looking at so they can look at it into more detail later. So they've pinned it down that's related to the endocytic pathway, and now that they know that, they can focus in further to see whether there's a viral synapse or something like that. Seeing like so I think this is just ruling out possibilities and narrowing things down. And so yeah, I'm not going to like talk down to this research just because I haven't done like some quick experiments I've just thought up on the fly. But I think this is still good. This is still good work. I think.
I think it totally is, and it's sort of interesting from that perspective of like what is like some of the maybe less talked about ways in which that infection could progress.
Yeah, and I think like if we're thinking about other sorts of issues like long-term COVID and other things, and then the idea that that SARS-CoV-2 can like transmit between
Yeah.
cells could be
I
an
think Minimal just
important
gave us
concept
like
for that.
Sigal. I guess that's this person isn't Segal. This is not the author. So some other scientist has also said that this might yeah, this might happen in the human lung. Yeah, not directly in vivo.
Yeah,
Yeah. Okay.
yeah. I'm anyway.
So
Yeah.
the next
So
paper that we
let's
have
go.
is the pigtail macaque. Model of COVID-19 reproduces diverse clinical outcomes and reveals new complex signatures of disease. So this is a new animal model. If people remember, some of the earliest papers that we saw from SARS-CoV-2 were done in the rhesus macaque, and
Hmm.
that particular primate cleared SARS-CoV-2 quite well. And so it wasn't really a good model if we're thinking about serious disease because yes, they get
Yeah.
sick and right they have the right receptors and they have primate biology, which is much closer to human biology. Um, now, oh, go
But
ahead.
yeah, no, I mean, it was, I think, a lot of trouble because I think we look at, I mean, our first Lost episode was where they were looking at
Remdesivir.
remdesivir, and it was very difficult for them to pick out when the primates had cleared it by themselves or whether they cleared it as a result of the
Exactly.
remdesivir.
And
And
that
so
was
it's
I think that
trying to
was
set,
also reported as, oh, this is great news for remdesivir, but then we saw in the human version, right? Like actually the therapeutic window for remdesivir is a little bit small. Hmm.
yeah, and like trying to focus in you because you're looking at when you've got an animal model which doesn't become sick, you're looking for just any difference, any kind of biomarkers that indicates that it might be something happening, and use that as your
Hmm.
proxy. And that makes things quite difficult because some of those things can be quite variable,
Hmm.
and so it would be nice to have a model that can more represent the human infection,
Yeah.
the human like situation.
Yeah. So I'm not sure if this is like almost too late to have a big impact, but it is very interesting to see that the choice of a different model, right, can give us like uh, a, an animal that has serious disease, that a primate animal that has serious disease, and it could be a very, I mean, maybe for future thoughts of like if coronaviruses again become an emergent virus, like it would be good to focus on maybe this particular species rather than another because of the way that it recapitulates disease. And sorry, and specific, I keep saying like this recapitulates disease, but what does that really mean? It's like it's all about that immune infiltrate in the lung. I believe that's how they're tying it back.
Mm.
That in these pigtail macaques, like they just get a lot. Their lungs are not in a good state after infection.
Yeah, yeah, you're getting like lots of. So I'm guessing that. So I'm trying to remember like hit my histology because I remember like the red stuff it tends to be the pinkish stuff tends to be blood and the dark purple stuff tends to be immune cells,
Yeah.
and you
Yeah, the
get
dark purple things, those
and
are because that's like the nuclei stain in the H&E. Yeah, then you're
yeah,
seeing like a lot of these dark things. Yeah.
yeah, and the structure of like because when you get lots of immune cells they can like bust through and so when the immune cells get into a place they have to like kind of squeeze past other cells and they can cause damage in the cells
Hmm.
in that way, so that can cause some changes to the structure of the area that's being looked
Hmm.
at. So I mean, and of course in pulmonary, in like the lungs you also get thickening of like certain areas as well
Hmm.
if because there's so much immune cells, but yeah um
Totally.
so yeah, lots of like pathology stuff in
Yep.
here.
And then also from the
Um,
blood counts, they just look at like the inflammation response. And again, they're seeing like these markers of like a more serious disease forming. And that is what's bringing them to this conclusion this may be a better model to study serious disease from coronaviruses in. Well, SARS-CoV-2, but I
mm,
I would think that the utility might be for because I'm not sure how much like we're going to need these like sort of in-depth SARS-CoV-2 models going forward, but like perhaps like it's like something about coronaviruses that this could speak towards.
yeah, potentially if you look at like really niche immune questions,
Yeah.
uh, or even if we get to a point where there is a new coronavirus where we don't have a vaccine candidate already,
Yes.
so
Yes.
or for whatever reason isn't ethical to experiment on
Right.
humans, then maybe this could be a useful starting point.
Yeah. Just trying to load up the
But
figure
yeah,
here to say something about it. Anyways, yeah, they look at T cell
which figure are you
responses.
looking for?
Oh, figure six. Oh yeah, it's T. They're looking at T cells in figure six. Yeah, so they're really zooming in. They zoom in very deeply. They're doing like single cell analysis of the whole T cell population and really
Right.
finding those markers that would say like, yeah, these infections are not going well for the animals, and but that's good when we're thinking about modeling a disease that doesn't go well for us.
Yeah, and I think I should point out that like even though these primates seem to have a much more severe disease than others, they haven't got like the most severe. So it's not like these primates were like getting extremely severe COVID that we've been seeing in humans.
Right.
But there is enough of a difference there for that you can potentially learn
Yeah.
something from the immune
Yeah, more
responses.
so than in the rhesus macaques. And I think that the other thing is like when you think about animal studies, like they don't like then try to put these animals on ventilators and like send them off, right? Like they get
Hmm.
euthanized at a certain point, right? And then yeah, because that I think is from
Yeah.
like the ethical standpoint, right? Like they are not extending the suffering of these animals, right? Like but at that point of serious disease, they can be euthanized, and then those tissues can be studied to get some molecular insight into what the immune system's doing at that point.
Yeah. I think like with primates, you kind of as long as you've got a good reason for euthanizing the animals, I think
Mm-hmm.
that's. But as far as I can tell, I think things are a bit more like there are a lot more regulations when you have to work with
Right.
primates than with other animals. So then I don't think so. Um, so I think this could be useful, but I think we've seen like the primate models kind of drop down like in use during
Yeah.
this for
Yeah.
SARS-CoV-2 as like better mouse models and hamster models have come
Right.
through.
Right.
Um,
Yeah, that's true. Yeah,
but
there's
I
still that fact that the humanized mice and the hamster models, they also recapitulate disease. So yeah.
but there is like I think one instance where primate models might be necessary is if you're studying neurological symptoms in long COVID because the
Mm,
way that primate brain is set up is quite different from rodent
sure.
brains. So that could potentially be one reason why this could be a useful
Yeah,
thing.
yeah, and that would also, right, like the brain. There's also crosstalk, right, in infection and the immune system. So it doesn't, yeah, this the increased relevance of this immune system to ours, yeah, may be helpful there in that particular instance.
Yeah, and I think that brings us to the end of our COVID
Mm-hmm.
papers. Um,
Yep.
yeah, and we're going to be talking about some other microbiology stuff.
Okay.
So,
First one on this docket is bacteriostatic antibiotics promote CRISPR-Cas adaptive immunity by enabling increased spacer acquisition. And I was quite excited about this paper because it's a model of um, immunity in bacteria. So they have a Pseudomonas that I guess is a few steps away from acquiring phage resistance through yeah, a few steps away from phage resistance through CRISPR, and it's I guess because there's some process right of genome change that has to happen for the adaptive immune right. CRISPR can also be thought of as an adaptive immune system for bacteria, and it requires changes to the genome in order to become fully operational. And so they have this model of a Pseudomonas strain that has part of its CRISPR-Cas system is ready to become resistant to a specific phage, DMS3-Vir, and in this particular study, they're studying whether or not if they treat that bacteria with a bacteriostatic antibiotic, which doesn't outright kill a bacteria, right? It just sort of puts the brakes on its growth. Whether or not that gives like a window of time for which it can better evolve phage resistance.
yeah, no, I think this is like quite it's quite interesting because they start off looking at like the effect of antibiotics on fate because I think antibiotics are getting leaked out into the soil like microbiome all the
Mm-hmm.
time. So I think this is important for environmental like trying to figure out what's happening out there. Um, and yeah, it's quite interesting the way they go because they like start off by looking at antibiotics in general and then they find that there's some specific mutations that happen with bacteriostatic antibiotics that don't necessarily happen with bactericidal. I think
Yeah.
they find that there's more spacer acquisition. So that's a chunk of viral DNA that they incorporate into the CRISPR system that can be used to target that virus in
So
future.
they
Um,
specifically zoom in, I think, on chloramphenicol in this paper, and that's an
mm,
antibiotic that sort of prevents proteins from being made. It messes up some ribosome stuff, but I guess it does it in a way that the cells don't explicitly die. And the readout that they're looking at is just the resistance to this phage that they have. And they find that when they're using these such as when they use antibiotics such as chloramphenicol, the bacteria grows slower, right? Especially if it's at like in sort of inhibitory concentrations, but sub maximum inhibition, right? Like it's preventing growth, but at the same time, those slower growing cultures are acquiring resistance to phages, and so a larger proportion of the bacteria in those cultures are phage resistant. And I think the reason why this is interesting for the authors is because we talked about this before with phage therapy, right? Is that maybe there's
mm,
some interplay between like being there's an interplay between using phages and using antibiotics where we want to use them like synergistically together in order to reduce
mm.
infections. And so understanding this relationship that slower growing bacteria are maybe more resistant to phages, that is going to inform the way that we do those types of combination therapies.
Yeah, and also like it's also interesting is how they figure out what's going
Mm-hmm.
on, because after they test out the bacteriostatic antibiotics, they test out different hypotheses, and then they take out the antibiotics entirely, and they give them like a carbon source that causes them to grow slower,
Mm-hmm.
and use that as a comparison. And that's how they found that just by making the bacteria grow slower on their own, that is enough for the phages for them to develop resistance much faster to the
Right,
phages,
right.
which I think was quite a neat way of like showing that this is the mechanism that by this is happening.
Mm-hmm.
So it's almost telling a complete story.
Yeah, absolutely. Actually, like I think that it's really it's quite um, it's like an interesting story is in dynamics, right? Because it's very much about the speed at which these things are happening, and but like scaled to I guess the lifetime of a bacteria and the population that experiences antibiotics and potentially a phage that is well, we hope that would be designed to like eliminate it.
I mean, it's interesting because like essentially what you're doing is like the phage is taking advantage of the bacteria's systems to try and
Hmm.
replicate itself fast. But once you slow all those systems
Yeah.
down, suddenly the phage is sort of growing slower, but the CRISPR is acting somewhat independently of that. It's
Yes.
still like even though the bacteria is stressed, it's still doing its job, and so it's almost like I guess that scene in the X-Men where like the guy goes really Quicksilver just like does things really fast and everything to him looks like it's going in slow motion. That's basically the CRISPR in this situation.
Yeah. I think I think it's a watch out too. Like that's an important piece of this information is
Mm.
like when we're designing these maybe combination therapies, right? Like we have to be aware that yeah, antibiotics are not always going to have like a synergistic beneficial effect, right? Depends on those mechanisms of action.
Mm. Yeah, we because now we kind of know that if we have partner up a phage
Yeah.
with like
Yeah.
chloramphenicol, that they might start working against each
Uh-huh.
other.
Uh-huh. Yeah, and then the other thing again that the reason I got kind of excited is like I didn't realize there were models, right? This is like a model system of a bacteria and its phage as a way of studying
Mm.
CRISPR immunity. And so, you know, we talk about model systems for studying human diseases, right? Where you have like animals and pathogens. Here we have like a model bacteria and its phage as a system to study these things. So I'm I'm excited probably to find more papers along this line as things go forward. It's cool to know that this work is being done.
Yeah, no, always interesting to see like how the science of phages is progressing, just because of background thinking of like phage
Hmm.
therapy or like environmental phages, and even like for the biotech aspects of phages. So there's lots of
Yeah.
like cool stuff going on here.
Yeah, and moving on. we have a bacterial membrane sculpting protein with bar domain-like activity. Um, so this is also maybe on the coattails of you being to that cell, the cell
Yeah,
conference. But these bar domains, they're well known in eukaryotic biology. They have specific like coils that interact with membranes and provide like a curvature force. So there's like a
yeah.
whole bunch of them, a whole bunch of examples in eukaryotic biology, but this paper finds an example in prokaryotic biology.
Yeah, and it's because we think of like endosomes as the circular things, but the endosomes have like a partner, which is like these nanotubes that can form. Because essentially, like you got these curvature proteins that can form like endosomes into spheres, but those curvature proteins can also like cause things to come all noodly and weird, uh, which um, which is quite a cool
Yeah,
like
I
thing
think
to see.
I
So
think
that
inside of eukaryotic biology, it's like a continuum, right? Like the Golgi apparatus is like this kind of blobby tubular thing, but off of it comes endosomes, right? So there's like a continuum based on something. I mean, I think bar proteins are contributing to that
yeah,
of like bending membranes into shapes that closely more get closer and closer to resembling just like little spheres, but you can have like all the intermediate shapes in between. And where you see those structures in bacteria is in these outer membrane vesicles that I think we've talked about them a little bit before, but like they're membrane protrusions that come out from some bacteria, and they also have a continuum of shape. In that, yeah, they're maybe start as tubes, and then at the ends they bleb off into actual vesicles.
right, yeah. And I think that there is like this idea of nanotubes as well. So of being used to like communicate between cells potentially, or because I know there's a thing for nets where like they throw out like a net of small like kind of tubes. Um, so I think that there is like more research being done into this sort of
Yeah,
area.
but
Um,
yeah, so this is very much like a discovery moment paper, right? And it was through direct observation. They have all these cryo-EM images and some fluorescence images of just looking at the production of these outer membrane vesicles, like sort of the tubes to the blebs, and they show that when they mutate out this putative bar domain BDPA, they get different shapes.
yeah, and they like see. So I think they've got some videos here, looking at like how it. Uh,
Yeah, they do have
so I
some
think
videos. They're not
so I
the
think this
easiest
is
ones to see. If you play one, it would be great, but I'm not sure if they're like super
yeah.
representative because they're looking like the bacteria are these big glowing things, but they're looking at like smaller chains that are kind of coming off of them. I just found it not easy to look
Yeah,
at.
yeah, it's quite difficult because I think the idea is like once this protein is overexpressed in some bacteria, it can cause nanotubes to form and kind of the membranes to start to go have this weird noodly kind of
Yeah.
look, and they actually transfer it from like the original bacteria they found, which was Shionella onai diendensis, and they go put it into E. coli, and let me see, they put it into another model organism. I can't remember which one it was
Yeah.
because I forgot.
Don't recall,
Yeah,
but it works in those bacteria as well. And then furthermore, like when you take now that they have this putative function of this protein, you can like align it to all bacterial proteins that we know, and actually, there might be other ones out there.
yeah,
Oh,
yeah,
yeah,
and
that's a good
that's
video that
quite
you
quite
just had.
helpful.
Yeah, you kind of see some of the
Yeah,
things growing out of it. Only two
so you got
seconds.
this clump. Yeah. So yeah, this is yeah, it's quite interesting to see how this all like play out.
Mhm.
Um, but I think that brings us to our final paper,
Sec-
which
oh, yeah.
is
Osmotrophy of dissolved
sorry,
organic carbon by coccolithophores in darkness. And so this is algae. So we we typically think of these as photosynthetic algae, but there's this. I guess there's an open question in people's view of deep history that if they were photosynthetic, but at some point, right, there was the the asteroid impact, right, created some dust layer, right, that like covered the Earth, and there was a period of some darkness. How did these guys survive if all they could do was be photosynthetic?
Yeah, it's an important thing because I think like because these coccoliths they can actually track them through the fossil record because they've got this like calcium kind of shell. Uh, so they're a big part of the nanofossil record. And I think what they found was like during like the Cretaceous like extinction, I think 90% of coccolithophore species were
Mm-hmm.
extinct. Um, so the question is how the 10% like survive? How do we still have them today if
Yes.
they mostly went
Yes, and so
extinct?
the hypothesis would be that they have some other way of incorporating carbon into themselves, and so they take some modern coccolithophores and they test them for this ability by, and we've seen this in some ecology papers before. You can use isotope labeling on specific things, and then you can then look. Is people think what's a good thought? I'm not sure if people are familiar with the carbon 14 dating, right? Where it's like you have a certain
Mm.
enrichment for a certain isotope of carbon, and then you can use that isotope sort of depletes over time, so you can use that to date something. Well, you can also just artificially induce that, introduce those isotopes into a culture, specifically on the carbon sources that you're interested in. So they have like a variety of different sugars that they have carbon 14 versions of. They introduce that into the culture under darkness, and they say, do these organisms grow, and do they incorporate those isotopes into their actual proteins and cells? And in some species they do, but not in all.
Yeah, so essentially if they didn't do anything, if they were literally just like not eating at all because they can't photosynthesize, these organisms wouldn't come out radioactive. But if they are eating these radioactive
Mm-hmm,
sugars around, then these organisms you'll find that they turn out
mm-hmm.
radioactive. So they found that this was what was happening. So through that they kind of like make had this idea that I think they kept these like cultures in complete darkness for about 30
Yep.
days and measured like how many cell divisions potentially they might have done. I think they accomplished about one cell division, and
It's very
so
slow.
this is very slow,
Uh-huh.
but it's still alive and still is doing metabolism,
Mm-hmm.
which is uh
It's just yeah, it's like a
yeah,
cool way
kind
of
of
uncovering, I guess, a metabolism that was inferred from was inferred from the fossil record, right? Like to know that they probably had to do something, and then following up with the living ancestors and saying, oh yeah, like they do have a strange, slow metabolism in this format.
yeah. So it is an interesting finding because I think they tend to think the coccolithophores are primarily photosynthetic, but the idea that they can have multiple strategies is kind of a new thing. It's like finding out like oh when you keep your ficus in darkness for too long, it'll start eating you. I mean that's kind of the
Yeah, a direct evidence for man-eating fungus.
yeah. I don't know why I thought of ficus was my first thing, but
or the man-eating part.
I don't know why my mind jumps to different places. I mean, I was partly thinking of like, don't look up for a
I
bit.
see.
Unfortunately, like, oh, some of these calculators actually listen to Leonardo DiCaprio and Jennifer Lawrence. But
But I
um,
think it's like again not super in a lot of science isn't driven by a lot of science is driven by application, but there's also science that's driven just by like
hmm,
trying to answer some very interested interesting questions about the history or like the way that life works. And this particular study does show us. I don't know if people have seen pictures. You should also Google coccolithophores. They're amazing under the SEM. And they're just like, yeah, it's just like a very fascinating life form, and the fact that they don't necessarily need to photosynthesize to survive, but have some backup metabolism. That's really cool.
Yeah, and we found this out through curiosity, but there are some applications for this. If you want to like grow these in culture, or I mean, coccolithophores, they sequester carbon. I mean, there's a lot
Yeah.
of other like things that people can think about when they.
Well, I guess the other
So
thing that I might say is that we don't know if we then investigate this particular pathway of coccolithophores using dissolved carbon sources as their primary mode of, right, of, or as a mode of growth. We don't know what we would find out if we understand the molecular mechanisms a bit better. And then from the biotech perspective, right, this is like the prospecting thing, right? Like these
right.
types of understanding a pathway could then be used, right, to engineer something that we would want. But yeah, I mean, to me, it's sort of like equal. I don't really know which way is better to go about science, right? Like I sort of people take different approaches, and we sort of hope that in the long run, like we'll find things that are useful, or just things that change the way that we tell stories about the world around us.
Yeah, I mean, yeah, whatever gets you grant funded. I mean, that's my motto.
Okay, so that's it for today's
Two
episode.
sides of science there.
That's it for today's episode. Join us next week for more cool microbiology news.
Yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the
Yeah,
comments.
I totally agree. Like, especially thank you to those in the comments who are adding their little tidbits. We really appreciate it. I did want to call it, like, I wasn't even thinking about, like, other organ systems, right, in primates that could be useful models. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process which we can all participate in, and we hope that you've had a good time listening to us ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny, and also thank you to all our brilliant
Yes,
commenters.
same here, Fuzz.
Tune in next week for more micro.